Section 9.3 is all about viruses. And so as we get started, we want to make sure that we know what a virus is and then how a virus works and what is the impact on humans or other organisms. When we first start talking about viruses, we do want to know that viruses are considered microorganisms. Micro meaning small, except they're really not considered alive. So there's a quote about viruses by a Nobel Prize winner that says viruses are a piece of bad news wrapped up in a protein. So we're going to see if we can understand where this saying came from. Viruses are not considered alive. It's this big debate. Are they alive? Are they not alive? They are grouped with microorganisms.
Charles Robinson, meet your ride in the front office. Charles Robinson, meet your ride in the front office. Teachers, if he's with you, please call the front office.
Mainly because they can cause disease. So when we look at microorganisms and the immune system, microorganisms are something that can cause disease. Keep in mind that it was once believed that RNA might have been the first nucleic acid, and so there is the question of, did viruses evolve into life? Did life backwards evolve? So let's look a little bit about what we mean. Viruses are not cellular. So they don't have a cell membrane, they don't have organelles, they don't have a ribosome, they don't have cytoplasm, they do have genetic material, and they do have an external structure. So the external structure of a cell would be the cell membrane or perhaps a cell wall. In viruses, it's a protein coat called a capsid. And this capsid can actually come in a variety of shapes. It can come helical, icosahedral, spherical, or these strange bacteriophages, which we'll talk about a little bit later. But the main idea is that these are actually proteins. And of course, looking at them, we can see that these shapes are actually going to be part of what we consider to be the antigen. So depending on the shape and depending on what's on the surface of that shape, we are going to see antigen variability. Because if it's a protein, it's actually coded for by something. So viruses basically have a protein coat called a capsid and a nucleic acid. That nucleic acid can be DNA, like we expect living things to all have DNA, or it can actually be RNA. Some viruses are double-stranded DNA. Some viruses are double-stranded RNA. Some viruses are single-stranded RNA. So we're used to seeing the genetic material be double-stranded DNA. But it turns out that some viruses only have RNA. We're going to talk about the implication. What does it mean if you only have RNA? We'll talk about that a little bit later. But the main thing to take away is that viruses do not have a cell membrane, and they vary in the forms of their genetic material. Now keep in mind, those genes are going to code for the proteins that the virus needs. So they have the gene, but they don't have any of the machinery. They don't have any of the enzymes. They can't do transcription and translation. One additional structure that some will have is called an envelope. And so an envelope is going to be phospholipids, and proteins, but this is from the host cell, and we'll see what that means later when we look down at our picture of HIV. But this is going to be an advantage because it helps avoid detection. It actually steals lipids and proteins from its host. As it bursts out of its host, it surrounds itself in its host cell's cell membrane and it helps it avoid detection from any immune responses.
Charles Robinson, please meet your ride in the front office. Charles Robinson, please meet your ride in the front office. Teachers, if he's with you, please call the front office.
So this is their structure. And then when we look at their function, really the only function that viruses have is to reproduce. Now it turns out that they can't reproduce on their own. They need a host cell. So they're what we call obligate intracellular parasites. They have to have a host cell to even be able to reproduce. We know that when we look at life, the main objective of life is to reproduce. That's all that viruses can do. They can't make toxins, they can't make anything other than their own structure, and they can't even do that on their own. Viruses actually replicate, copy, or reproduce in two different ways. The first is called the Lytic cycle. And when we mean two different ways, we don't mean that a virus can switch back and forth. Its genetic code programs it to either be a lytic virus or a lysogenic virus. A lytic virus, lice, is the root word here. And lice means to burst. The Lytic cycle really has four main steps. If we start with the step of a virus coming out and attaching. So the first step is attachment to host cell. And this sounds pretty simple, but we have to keep in mind that this attachment is usually going to require some sort of receptor. And we do want to indicate that usually it's very specific. So this is why if you get infected with a virus, your symptoms might be very localized. So the rhinovirus, or the common cold, is really just going to infect the upper respiratory system. Versus if you have pneumonia influenzae, you're going to have infection of the lower respiratory system, such as the lungs. So we do want to understand that we are cellular specific. So the virus has to be able to attach to another cell. Its next step is to inject its nucleic acid. Now keep in mind, the capsid is going to stay on the outside. Remember, this was how Hershey and Chase proved that the hereditary information was not the protein, that what you inherit is actually the nucleic acid. And so it injects its nucleic acid. It's going to hijack the cell's machinery to replicate its nucleic acid, and synthesize proteins. So that is really the third step. Now keep in mind, these are very simplified steps. It is not quite this straightforward, but these are the main processes that we see. And of course, as you start to look at HIV specifically and ways to inhibit viruses, we are going to see that each of those steps is really a pathway where we could stop viral replication. The fourth step is actually going to be the swelling and the lysing of the host cell. And this is necessary for the next infection. And so one of the components that we'll actually see as part of this is that somehow the virus needs to get not only from cell to cell, but possibly even from host to host. So this is why the destruction of certain cells is going to trigger the immune system to sneeze. And as you sneeze, or cough, or vomit, you're going to expel some of those virus and be able to transmit we do have cell specificity, but it turns out that there's also host specificity. That just because it infects the cell of a human does not mean that it's going to infect the cell of every animal. And that's where we start to see emerging viruses and how viruses are sometimes contained to one species, but sometimes they jump or spread from species to species. One thing that we do need to keep in mind is that because the virus is not cellular, medications that target the cell wall or the ribosome are not going to have any effect on the virus. If we want to be able to stop a virus, we need to be able to stop the virus during its reproductive cycle, and we need to be able to identify all of those steps. Now this is the Lytic cycle. This is how some viruses are going to reproduce or replicate. Next we want to look at another group of viruses that are lysogenic. And lysogenic are also called temperate viruses, or sometimes they're called latent viruses. Now, lysogenic viruses are going to be viruses that have a period of dormancy. Which is where the viral DNA is incorporated into the host DNA. And what this means is that the virus is actually able to evade detection by staying in the host cell for an extended period of time. This period of dormancy means there may be no reproduction of the virus for an extended period of time. And then some environmental trigger will push the virus into the Lytic cycle, will result in the Lytic cycle. So lysogenic viruses are going to eventually finish the Lytic cycle. They're going to reproduce, replicate their nucleic acid, they're going to swell and burst the host cell, and then they're going to start the process all over again. But lysogenic viruses have a temperate, a latent, a period of dormancy where they're actually incorporated into the host cell. Now keep in mind, if I'm incorporated into the host's DNA, then every time that host cell divides, the viral DNA gets replicated as well. So it turns out that there are some bacteria that can get infected. Every single living thing on Earth can be infected with a virus. And so we do see a really high frequency of bacteria being infected with viruses. And what this means is that every time the virus reproduces, the bacteria reproduces, the viral DNA is replicated as well. And this is actually a source of variation in bacteria. And it is one way that bacterial genes are sometimes shared. Because when the virus eventually enters the Lytic cycle, sometimes it takes with it some bacterial DNA, and then when it infects the next bacteria, it's sharing those bacterial genes. One of the interesting things that we'll begin to talk about is the idea of a retrovirus, and a virus that can go from RNA, retro back to DNA, and then back to RNA. So most lysogenic viruses are also retroviruses, which we'll talk more about